Hello, AP Bio students. I hope you've enjoyed homecoming weekend. Uh, got chilly quite soon, so let's go ahead and continue with what we're learning. We left on the previous video with our accounting with glycolysis and Krebs. Now we haven't made that much ATP so far for ATP, and that's been with substrate level phosphorylation, which is not the most efficient. You can see after all of these reactions, we've only been able to tap into that to get for ATP. So how in the world do we get the rest? And so before we get to this, and by the way, this is what it's going to look like. Here's our membrane. And where this is coming from, let me show you a little diagram. Here's our mitochondria. And the mitochondria has this weird folded membrane, which we'll kind of talk about. And imagine, if you will, let me go ahead and highlight. This little layer right here is pointing to this inner membrane. And so just to clarify, I'm going to go ahead and use the orange. The orange would be this intermembrane space right here. Now, I would suggest I'm using different colors. That really does help. And we're going to practice this a little bit on Tuesday as well. But that's the intermembrane space. And then just to continue this, the yellow would be the matrix. And so you can see the colors match where they are as well. So that'll give you some perspective of where this is. Now, we're looking at these proteins. The purple are proteins, but what in the world are these? So let's go back up to this graph. Now, I'm going to go ahead and suggest that you pause it at this point, and go ahead and write down the information in your notes. You have this in your notes from Friday, and so just take a second, pause it. And now that you have finished writing this down, you will be able to follow along much more comfortably. So on this graph, you have the free energy on the left-hand side. And this represents the amount of available energy. And notice it's going to go high to low. So from 50, 40, 30, 20, 10, and 0. Okay, so that shows you the energy, and notice it's going down these molecules. So this picture is much more like a graph than it is an actual representation. So the reactions are going from left to right, and you're seeing you're seeing the measurement of the energy. On the right-hand side, now your book doesn't have this, but I think it's actually worthwhile to go over. What you have on the right-hand side would be a measurement of the redox potential. And the redox potential is a measure of how willing these molecules are to give or take electrons. So up here on the top, it would be negative 400, which is a measure of how willing it is to donate. These molecules are very willing to give their electrons. Down here on the bottom, it would be 800. That's the most positive redox potential. And this represents it being most willing to accept. It's the high electronegativity. And notice we're looking at oxygen, which has both the lowest energy and also the highest redox potential. So that's kind of an explanation of this graph. Now, what you're looking at are all of these molecules that are placed together in an arrangement based on that energy and redox potential. So if I could make mine a little messy, what we're going to see is a electron. Actually, let me use the yellow. This electron gets donated to this FMN, the flavomononucleide and mononucleotide, sorry, and it's going to gain that electron. Now remember, gaining electron means that it is reduced, and then this is going to then donate its electron to iron sulfur, and then so the FMN is now oxidized as it loses that electron, and now the iron sulfur is reduced. And that's going to happen, so forth, and so on, and so on, and so on. And notice as this is happening, the electron drops both in energy and then the molecules increase in their electronegativity. So it would be similar to you being in a line where the last person in the line is just gimme, gimme, gimme that electron. They want that electron. Whereas the first person in the line is very willing. I'm going to pass it down. And because that interesting relationship between willingness to give and ease of taking, it becomes very easy, just like a little slinky going down the stairs. This electron just naturally falls from molecule to molecule. Now, each time that these electrons hit the molecules, that energy gives that capability of doing something, and we're going to see that in just a bit. Now, what in the world are these things, these weird letters? Well, the purple are proteins, and within the proteins you have prosthetic groups. Now, we actually just talked about this last week. Prosthetic groups are permanent coenzymes and cofactors. And we talked about these. These are molecules, most likely they're either organic, like vitamin based. For instance, the first one has flavin as part of its name, and you might maybe, if you've ever heard of some of the vitamins like riboflavin, and so that might help you think about why those vitamins are so important. They actually become part of our metabolic processes. And then notice a lot of the other ones are metals: iron, sulfur, iron, sulfur, iron, sulfur. And then this quinone, and which that doesn't tell you much, but it's a molecule that has an easy transition from one electron to the next electron. And then we have this B, C1, C, A, and A3, and those are all cytochromes. And cytochrome has an iron group in it called a heme group. Now, some of you from anatomy might remember that's actually in red blood cells, the hemoglobin. In the hemoglobin, it attaches oxygen. In this case, it's going to grab electrons. All right, so what? So what in the world, why do we care? What about this? And then notice, by the way, down here, the oxygen is going to be the one that's grabbing it. That's the final electron acceptor. And so again, just to kind of bring it home to a take-home message, we have these successive molecules that are increasing in electronegativity, which make it natural for the electron to kind of just bounce from one molecule to the next. So why do we care? Well, let's go ahead and get to this diagram. Now, this diagram shows complex 1, complex 2, 3. Well, here we have the cytochrome C, and then complex 4. Now, these molecules, as they're gaining that electron, and again, just to kind of connect it to what we just saw, here's our electron, and the electron's bouncing from one to the next, to the next, to the next, to the next, and then finally to oxygen to become water. Okay, so let's really drive home what's happening. Here's where the big payoff is. What can happen when these electrons are being given to these molecules? They have that potential energy in them, and they can join with a proton to pass through. That proton can then be passed through to the other side. So this is actually pumping. So this is a form of active transport. So this is using that energy to take protons from the inside of the matrix to the intermembrane space. So they're being dragged over this membrane. Same here, same here. All right, and this continually happens because if we go back to the very beginning, once these NADHs are done dropping these off, they're going to go back and pick up. Remember, they're dump trucks. That's all they do. They dump off the electron and then go back to pick up more. So the Krebs cycle, we really see it. So this is going to go off to the electron transport system, dump off its electron, and then it's going to circle back. Same thing with all of these. So as long as you are still alive, your electron transport molecules are happily just cycling back and forth. So just to recap, you're going to have a Krebs cycle that's occurring down here. And of course, it's not just one, right? You have a Krebs cycle over here that's happening and a Krebs cycle over here. And so these NADH, they're dropping off their electrons and then coming back, grabbing another electron and then going back. And so you just get these shuttling of electrons back and forth. And so as this keeps happening, you've got another electron that's being put in and then another hydrogen that can be pumped through. And then it goes to pick up another electron, comes back, goes through, another hydrogen ion goes through. And then you just have this cycling back and forth. And as it keeps going, you're pumping more hydrogen ions through. All right, well, that's well and good. And this can happen a couple more times. So we can move our hydrogen ions across in another pumping. And hopefully you're starting to make a observation in your mind that as we continue to add these hydrogen ions, well, lo and behold, this is now a high concentration of hydrogens. And that means something, because if you have a high concentration, that high concentration will naturally move to a low concentration if you allow it. Now, just as a reminder, these are charged ions. Well, they're charged atoms, they're ions, and they can't move through those nonpolar tails, so they bounce off. But if given the chance, they can go through a transport. Now, this transporter is a very unique transporter. It's ATP synthase. And that ATP synthase is very similar to a proton pump, but this time it's working in reverse. So instead of taking ATP to move protons across, the protons are going to move through it and drive that molecule to connect ADP and phosphate. So it's this chemiosmosis, and so that's the word they kind of used here, the chemi. So like chemical osmosis, now that's kind of erroneous. That implies water, but it's using that diffusion idea. So chemiosmosis, which is using that proton gradient to then convert ADP into and phosphate into ATP. And then this just keeps happening and happening and happening and happening. Now I want you to think about a couple of things. I want you to think about, what if we had a molecule that unfortunately made a hole through this membrane? So that's scenario one. And then I want you to think of a couple of other scenarios. What if we then had another molecule that could block this ATP synthase? So that's scenario two. And then what if we had another molecule that, instead of the NADH making donating the electron to complex 1, what if we had a molecule that kind of snatched that electron away? So I want you to think and see if you can predict what would happen with each of these scenarios. Now, the other thing I would like you to do is go ahead and see how well, just by like memory, and of course, if you can't, you can go back to the beginning page, how well you kind of know what's going on in each of these boxes. This is in chapter 9 of your book, so by all means, open up your book and take some notes and make sure you figure that out. And then we'll finish up with some practice and application on Tuesday. So hope this helps get you thinking about everything. Remember the ideas that we went over today. We looked at how the electron transport system is kind of built and how it works together to allow the electrons to drop down in energy, and then how that energy is used to then make ATP. And then I'd like you to try to put all of these things together, and then we'll talk about this on Tuesday. So have a lovely weekend.